So today's speaker is Peter Adolfatto, who comes to us from Princeton University. He got his undergraduate degree at Simon Fraser Institute University in Canada. And you'll hear when he starts to speak that he is in fact Canadian. and then he did his PhD at the University of Chicago with Marty Kreitman, and that's where I first met Peter when he was a graduate student. And I remember being very impressed then, and his career has just blossomed. From there he went to he did a postdoc at Edinburgh, and then took his first faculty position at the University of Toronto. He's been a little bit peripatetic, and then he went to UC San Diego for, what, 3 or 4 years? 4 years, before going to Princeton University, where he's been for 6 years and is now an associate professor. So he was an assistant professor for 10 years because of his moving around all the time. But during that time, he's been enormously productive. And what's interesting about Peter is he has interests on a number of very different fronts. And so his research has included things like detecting selection in patterns of polymorphism and divergence in Drosophila. Most notably, a paper was very influential, published in Nature in 2005, showing that there seems to be a lot of strong directional selection in non-coding regions in Drosophila, to things like speciation in swordtail fish, which he's not going to talk about today, but is some interesting ongoing work in his lab. But today's talk is going to be all about herbivores that feed on toxic plants and related things. And he tells me it's not just going to be about plants and insects, but there will be some vertebrates in today's talk as well. The last thing I want to say is if you are interested in meeting with him and weren't able to get on his schedule, he has a little time tomorrow morning. So if you'd like to meet with him, please send me an email. So Peter, thank you for coming.
Yeah, thanks for inviting me. Is there a pointer, by the way? That you guys
It's in the top right-hand drawer. That black.
This thing?
Yeah.
Oh yeah, there you go. Yeah, so I came across this article in a paper recently. Scientists claim humans could evolve beaks instead of teeth, and with the important caveat, but not for millions of years. Why did I think this was important? Well, I thought this was a great segue to the talk, because to me this brings up what I think are two fundamental but unanswered questions in evolutionary biology, and that is what factors limit the rate of adaptive evolution, and to what extent are evolutionary paths predictable? So there are a number of ideas about answers to these questions. The first, and perhaps the simplest, is that this just largely depends on the size of the mutational target. You can see in the case where there's just one mutation, one possible mutation will bring you to, will accomplish the job. You see that adaptation will both be maximally limited and maximally predictable. So that's not that interesting. The question is, how many traits that distinguish populations or species really have such a simple genetic architecture? Probably not very many. The second idea out there is this idea that perhaps evolutionary paths are epistatic. And so the way this works is that perhaps a newly arising mutation is only beneficial if it occurs on the right genetic background. Now it's just a matter of waiting for that right genetic background to arise. And this is both going to limit the rate of adaptive evolution and make it more predictable than it would be otherwise. And then a third kind of major stream of thinking out there is that this has to do with negative pleiotropy. And that is, perhaps many possibly otherwise beneficial mutations are actually associated with indirect deleterious consequences, such that they never have a chance of really fixing the population. And so superficially, this looks a lot like just a small mutation target size, but it's quite different in the sense that if something changes to release those negative pleiotropic constraints, then the rate of adaptive evolution can speed up quite a bit, and evolutionary paths can become quite unpredictable. So how do we go about distinguishing among those factors? That's particularly difficult because the evolutionary process for any particular trait has only happened once. And so one approach is to do a literature review and cobble together as many examples of adaptations that dissected to the molecular level that you can, as was done in these two reviews. But the difficulty here is that you're left comparing adaptations that occurred for very different traits in different contexts, so it's unclear to what extent you can really make generalizations. What you would ideally like to do instead is run the evolutionary process over and over again for the same trait. So the right idea would be some sort of chemostat experiment. And I'm citing a recent, particularly great example of one of these experiments. And this is quite a powerful approach. However, it does suffer from its own limitations. The first being, well, these are largely only applicable to microbes, which you might not think will be representative of a lot of taxa, particularly if you're talking in a museum of vertebrate zoology. But I think the bigger problem here is that the strength of selection imposed in these chemostat experiments is actually quite strong. So if there were negative pleiotropic constraints attached to some of these beneficial mutations, selection might be strong enough to cause their fixation anyway. And then a third point is, related to the second one, is that the environmental conditions in these chemostats are quite a simplification of nature, and so that could in turn cover up hidden negative pleiotropic effects. They're not necessarily representative of the type of adaptive substitutions that will happen actually in nature. So how do we approach this then? An alternative approach is to consider examples of convergent evolution. And so convergent evolution is not only great evidence that evolution is true, but given that you understand enough about the genetic architecture of the trait that you're looking at, it's a potentially powerful tool to learn about the genetics of adaptation. So in that regard, this is a great example of convergent evolution, independent evolution of the wing, but it's actually not the best trait to pick if you would like to understand the genetic underpinnings of the trait. And so I think the benchmark for this kind of study, I mean, there are an accumulating number of studies where the genetic basis of convergent evolution has been dissected to the molecular level. And I think the benchmark in one of the first studies is this really nice work done by Yokoyama and colleagues on red-green visual pigments of opsins in vertebrates. And I think Yokoyama and colleagues really set the benchmark for how such studies should go. What they found is that what they notice is recurrent substitutions to the same 5 sites in these opsins causes a shift from red opsins to green opsins. You see this happening recurrently 4 independent times, and then supporting functional studies show that variation at these 5 sites can just account for all the phenotypic variation in invertebrates. And what they see is a high degree of parallelism at the molecular level. These 3 substitutions, for example, occur 3 times independently in the tree, shifting things from predictably from red to green. And you also see recurrent duplications that facilitate dichromatic vision in cave fish and in humans. Also in this example, he sees epistasis. So for example, once you start down the path by changing this histidine to a tyrosine at position 197, now it turns out that none of these mutations will actually bring you further down the road to shift you towards green. Instead, you're stuck having to incorporate this alternative substitution. Inspired by this example, I'd say the most informative examples of this type of study should be those that examine outcomes of natural rather than artificial selection, those that are able to integrate evolutionary and functional analyses, and then those that can survey a large number of independent outcomes under a common selective pressure. And so clearly, the larger number of outcomes, the more independent trials of evolution you have, the better it is. But you also would like the selection pressure to be as common as possible. And if we go back to that Yokoyama study, it's a bit unclear that these shifts are really all the same selective pressure. It'd be nice to have more examples of this kind of thing. And so the example I'm going to talk about today is concerns Apocynaceae plants and their herbivores. Apocynaceae plants, as a defense against herbivory, among other adaptations, produce a class of toxic compounds called cardenolides, which all have this kind of, this is a representative one, they all have this classic steroid core connected to a sugar, and then a lactone group out here. Cardenolides have a specific target, an essential gene in all animals called sodium-potassium ATPase, which is responsible for generating sodium-potassium gradients, driving a lot of cellular processes. So everything from osmoregulation to muscle contraction to neural signal transduction, among many other processes. This is an essential enzyme. And so the existence of a binding site for this compound is actually no accident. It's actually thought that animals synthesize compounds that look a lot like this, perhaps even this one, to regulate the activity of the pump endogenously. So this is a normal regulatory interaction in all animals that plants are exploiting. That's the nature of the toxin. And in fact, we've known about this for quite a bit of time, because for hundreds of years, actually, we've been using plant extracts to treat conditions like cardiac arrhythmias and congestive heart failure using this low doses of these compounds. And it's because of its medical importance that we know a lot about this protein, and we know specifically a lot about the interaction of this specific ligand with the protein, including very nice co-crystal structures of for example, in this case, the pig protein bound to a generic cardenolide. And then in addition to this, quite a wealth of direct functional studies that have documented by site-specific mutagenesis or protein crystal structure studies residues throughout the entire protein at which substitutions can modulate the enzyme's affinity for this toxin. And the take-home picture here is that we're not just talking about a small mutational target here. It's something like at least 35 sites widely scattered throughout the entire protein have the potential to confer resistance to cardenolide inhibition. And so I also want to emphasize that this target site insensitivity is just one of multiple routes to cardenolide insensitivity. It actually turns out that some animals are endowed with extra protection. For example, if you took tons of this toxin and you fed it to Drosophila melanogaster, it would be just fine. And first that surprised me, but then I thought, well, actually this makes sense because it's a generalist that lives in garbage cans. It should probably encounter these compounds like this a lot. In fact, Drosophila melanogaster and probably a lot of other insects co-express these generic ABC transporters with sodium-potassium ATPase. They latch onto things that look like the toxin and just excrete it immediately before it can do any damage. And then yet a third strategy has been documented, particularly in a lot of Lepidoptera, but I'm sure other species go this route as well. What you do is you restrict the expression of sodium-potassium ATPase to the nervous system where its role is the most crucial. You use other systems to move sodium-potassium around in other tissues. neurons are protected by a perineurium blood-brain barrier that protects you from polar versions of this toxin, and non-polar versions of this toxin can still leak through. They have this added protection of these phosphorylated glycoproteins, which act a lot like these ABC transporters to latch onto these things and excrete them. So this is by no means a small mutational target. And so despite the strategy of these plants in deterring herbivores by producing these toxins, when you look closely at the plants, you see nonetheless a lot of species from a lot of insect orders have the ability to feed on this plant, and they're happy to chew away on this plant, but they actually take it a step further and they sequester the toxins for use in their own defense. And you can see a lot of these species have co-evolved warning coloration, advertising their unpalatability. And so here I'm showing, with the exception of 3 butterflies here, all of these species can be found basically in my backyard in New Jersey. But worldwide, it's estimated that something like on the order of several hundred insect species have the ability to do this on this family of plants. And so it's the large number of taxa involved here, combined with the detailed knowledge of the interaction between sodium potassium ATPase and this ligand that attracted me to this system, and I think provides a really cool opportunity to look at the potential here for convergent evolution. And so looking back at this site of potential target sites in the protein, the question to an evolutionary biologist becomes, which of these sites are going to be used in the evolution of cardenolide insensitivity, and why those sites and not others. And so the challenge here was to sequence this rather large protein, 1,000 amino acids, in about 30 non-model species spanning 300 million years of evolution. So I actually kind of started this as a grad student doing degenerate PCR, and that's just not going to work. If you look at the level of DNA divergence between the roots of these trees, Lepidoptera to hemipterans here, it's even the coding sequence is something like 33% diverged. So it's just very highly divergent despite being a conserved protein. What we did, to make a long story short, we took just a brute force genomics approach. We just ground up each of these animals, prepared indexed RNA-seq libraries, sequenced them en masse, and then subjected those to de novo assembly and then a global alignment. And this is one of those slides that sums up months of work. And but the point of that was to produce a data set that looks like this. And so what I've done, instead of showing you all the sites in the protein, I just have the 35 sites that have the potential to modulate cardenolide sensitivity across the top here. I have a reference sequence up here. Out here are the vertebrates. No, these are not the only vertebrates in the top. And then here's a phylogeny relating all the insects we surveyed. In yellow are Apocynaceae plant users. In white are some informative outgroups. And in gray are casual users. It's kind of unclear that these species actually sequester cardenolides at all. And so in this data set, you see that, well, not all of these sites are being used in the 300 million plus. Actually, if you added up all the evolutionary time here, it would be 1 billion years of evolution. We see 49 substitutions at just 19 of the sites. 33 of those substitutions occur in Apocynaceae specialists. And about half of those 33, we actually, that specific mutation, and those are highlighted in red here, we know that that specific mutation has the ability, decreases the enzyme's affinity for cardenolide. And so the thing is, those functional studies have limitations. We're able to identify half of the substitution, we're able to attribute functional consequences to half of the substitutions. In order to get a better idea of the basis for cardenolide insensitivity, what we did was some protein structure modeling. And this is called ligand docking simulations. So the way this works is that we have the PEG structure, the co-crystal structure of the enzyme with the ligand. What we do is we take that structure, we extract the ligand, and then we let it re-dock and find the best docking location. And that's shown with the ligand in red here. And then what we do is just by computer add the mutation we want to the protein, we refold it, and then allow the ligand to dock again, and that's shown in blue here. And so this shows the ligand docking phenotype for this histidine substitution to asparagine 122. And as you can see, this mutation that it's known to have an effect on cardenolide sensitivity is sterically blocking the cardenolide from penetrating the binding pocket. And so this kind of thing helps us understand the molecular basis for insensitivity. We can look at the second substitution we know also affects cardenolide insensitivity. Interestingly, it's a very different substitution in a different part of the protein. When we do the ligand docking simulations, it produces a ligand docking phenotype that's very similar to this other substitution. And so those are substitutions with known effects. We then applied this same procedure to all substitutions to see if we could learn more. And what we found was that 3 substitutions here, this one, and this one, produce ligand docking simulation phenotypes that are very similar to these other 2 substitutions of known effect. So we've discovered new substitutions of known effect using this approach. In general, though, these were the only 3 we found. So this is not a particularly sensitive way to find these things. And so I think it's pretty clear that there's been accelerated functional protein evolution in Apocynaceae-associated taxa. The best way to see this is, let's ignore the phylogeny for now, just group outgroups together and specialists together, and whether you count all substitutions or those just with known effects, the enrichment among specialists is highly significant. This is adaptive protein evolution. Sites are used very not equally. Okay, so among the 35 potential sites, only 19 were used, but actually 2/3 of all the substitutions are just localized to this one extracellular loop in the protein. We see extensive parallelism at 2 sites in particular, and these flank the boundaries of that loop. At position 111, there are 12 substitutions, and all of them occur in parallel. So to get this straight, what I mean by parallel is that they occur in at least 2 independent evolutionary lineages. At the second most substituted site is at the other end of this extracellular loop. There are 6 substitutions, 5 of which occur in parallel, and 1 of which is unique. And interestingly, half of all the substitutions we detected happen at just these 2 sites. So is evolution predictable in this case? Well, yes, depending on how you want to see it. Glass half full is predictable because 50% of the sites, 50% of the substitutions occur at just 2 sites. That's amazing. Glass half empty, what accounts for the unique substitutions? So how do we explain the other substitutions? That actually turned out to be quite interesting as well. And the reason is because what we found is 3 independent rounds of duplication, of recent duplications of this sodium-potassium ATPase gene. And I say relatively recent because, as you can see, the youngest one is 9 million years old. The oldest one is 125 million years old. What this implies is that many species in that group are likely to share the same duplication. But I think understanding what goes on in the case of when you're duplicated versus not duplicated is really quite informative. So if we just now consider specialists and we consider those with a single copy versus those specialists that have a duplicated copy, what we see is that if you only have a single copy, evolution is largely limited to evolution at just these two sites, and they're parallel substitutions. Unique substitutions are rare. Whereas if you duplicate the copy, all of a sudden all bets are off and you start to see a lot more unique solutions to this evolutionary problem. And so this strongly suggested to us that yes, recurrently convergent solution of gene duplication has led to less predictability in evolution because of this release of negative pleiotropic effects. So some more evidence from that comes from considering gene expression patterns of the duplicates. So for example, in the case of these two beetles, here they have 2 copies each. They're very distantly related to one another. For each of those, based on the pattern of substitution, we have a prediction for which copy is more sensitive and which copy is more resistant. We actually have these docking simulations where we simulated the structure of the native protein. That includes all of the substitutions everywhere in the protein. And we confirmed that indeed the blue copy here looks like the resistant copy in the sense that it's displacing cardenolide. And you can see, even though the substitutions are very different in these 2 cases, the displacement phenotype is very similar in these 2 cases. But here's the expression data. What we looked at is the relative expression of the resistant copy versus the sensitive copy in 3 different tissues, the head, muscle, and the gut. And what we found in both cases is that the resistant copy is more highly expressed in the gut. The sensitive copy is more highly expressed in the brain. And that is a little bit satisfying, because in the gut, your first line of defense against a toxin, that's where you have to deal with the toxin first, and you're stuck with absorbing it through your gut. So you have to deal with it there. The neurons, on the other hand, are partially protected by the perineurium, and they also, they're performing a very crucial function at very short time scales. So you want that copy to be close to the ancestral best working copy of sodium potassium ATPase. That's a guess at the moment. We're trying to get proof, but that's a little ways off still. We also see this in the bugs. These are the hemipterans. In that case, it's a bit more complicated because there are 3 copies, but we can take these 3 copies and rank them as the most resistant looking one, the most sensitive looking one. They all have some degree of resistance to the toxin. But it's, again, always the case that if we compare a more resistant one to a more sensitive one, we see the more resistant one is more highly expressed in the gut relative to the head. And we see this in both species of milkweed bug. And that's not surprising, since the duplications here actually preceded the divergence of these species. But just to show independent data for 2 different species. So, in summary, we see this striking parallelism at the molecular level associated with the ability to sequester cardenolides. We see hotspots for parallel amino acid substitution, but we also see unique substitutions that are disproportionately associated with recent duplications of sodium potassium ATPase, and these duplications are differentially expressed in tissues. But together, I think this data is strongly support a role for negative pleiotropy in limiting the rate of adaptation and increasing predictability. I think the thing that's missing here is to actually show that there are trade-offs associated with, in particular, the unique substitutions. And we're working on that. So in terms of future directions, we're kind of stepping back and kind of looking more broadly at this as kind of a paradigm that could be applied more generally. And so there are plenty of situations where you see plants co-evolving with herbivores, herbivores co-evolving with natural enemies, and this being a rather large community subject to a similar selective pressure in the context of this chemical warfare, essentially. There are a number of examples. Sodium potassium ATPase and cardenolide is really just one of these examples. We made a lot of progress focusing on just this part of the graph of one system, basically. But it seems obvious. I told you that we were able to make really neat inferences from just surveying 14 species. But now we're in the process of, I mentioned that worldwide there's something like several hundred species doing the same thing. We could start to fill in our catalog of solutions here, and I think it's very promising that we're going to learn new stuff as we add new groups. And in particular, I'm pretty excited about, we've so far surveyed 3 insect orders. We're planning to add very soon these two additional insect orders. This is the dipteran, and these are orthopterans, grasshoppers. Another axis that we're expanding on this is we're starting to look at this part of the graph, specifically for sodium potassium ATPase and cardenolide, and that is these herbivores living on these plants. They themselves are toxic and they're preyed upon by natural enemies. These include a number of vertebrate and invertebrate predators. These, for example, are species preying on monarchs at overwintering sites in Mexico. Some of these predators have evolved behaviors to actually try to avoid the most toxic parts of the monarch, implying that this activity is exerting pressure on them, and they're paying a cost. And I think there's pressure for these predators to evolve resistance. I could show you also some invertebrate predators as well, but this also includes many of these herbivores are subject to parasites or parasitoids. For example, these tachinid flies actually do quite a bit of damage to Lepidoptera, and there are ones that specialize specifically on these milkweed feeding taxa. But it also includes mutualists, so this is quite funny. There are a number of aphid species living on these host plants that are farmed by aphid-tending ants. And basically these aphids release a honeydew that the ants are feeding on. The ones on Apocynaceae plants, the honeydew that they're making is laced with toxins, and it's unclear how the ants are coping with this. We're planning to take a look. Another axis we're expanding this study on is to look at analogous predator-prey systems. So it turns out that I just told you a story that was centered around Apocynaceae plants, which have evolved the ability to make a toxin, and that propagates up a trophic chain. There are actually 2 animal groups that have the ability to make compounds that look a lot like these cardenolides. So I maybe faded that out a bit, but here's our generic cardenolide, and this is a compound produced by this guy here, the buffonid toad, and this is a cane toad, actually, I guess. And so these guys actually, when threatened, will start to secrete these toxins to deter predators. And these buffonid frogs are preyed upon by snakes, lizards, and predatory frogs. So I think you have a situation here where you can look at an entire community of organisms all trying to deal with the same evolutionary problem. These guys have to evolve resistance to the toxin. These guys have to evolve resistance to the toxin because they're eating that guy. These snakes actually sequester toxins from eating these frogs. And so a little bit of preliminary work has been done on this. This is a paper that came out recently by Moore et al. In Biology Letters. What they looked at was the evolution of sodium potassium ATPase. They used a degenerate primer PCR approach and just looked at that first extracellular loop. And they looked at it in a number of buffonid frogs here and one predator, which happens to be this Leptodactylus species. And it's actually quite interesting. What they found was what looks like a recent duplication of sodium potassium ATPase, mirroring what we saw in the insects. And actually they see a number of amino acid substitutions here. I highlighted these because these are known to have functional effects based on the functional data. But you see this pattern reminiscent of what we saw in the insects, and that is they've modified one copy and kept the other copy constant. And so in collaboration with Andrew Crawford, who's doing a sabbatical visit here, and his graduate student Pilar, we are looking at the evolution of sodium potassium ATPase in the context of a broader sample of frogs. And instead of just looking at the extracellular loop, looking at the entire protein and trying to do this in more detail, hopefully adding some of the other vertebrate predators into the system. But another place we've made progress is on these guys. I mentioned there were two animal groups that have evolved the ability to make cardenolide-like compounds. This is the second one. This is a picture of a park in Princeton around mid-June, and those are fireflies. Turns out that the fireflies in New Jersey, Pennsylvania, and New York are unusual as fireflies goes because they happen to be day active. And then you see actually they come with this warning coloration advertising unpalatability. And it turns out they're rich in these compounds. This one's called lucibuphigin. It looks almost exactly like the toad toxin, and it's pretty, which is pretty close to this compound made by the plants. There are two genera involved: Photinus, which has the ability to make these compounds, and then Photurus, which actually lacks the ability to make these compounds, but does have the ability to eat this other firefly. And you see there's quite a size differential there. And actually there's quite an interesting story in the sense that this is a Photinus female. The Photinus female, the way she catches, Photurus, female, the way she catches Photinus males is she hides in the grass and mimics the flash of females of Photinus species, sucking males in, and then she chomps on them. And we can actually catch them by watching that dynamic play out. So applying an approach that's just very similar to how we dealt with the plant herbivore system, we just ground up a bunch of these guys and threw them onto the sequencer, and then cataloged all the variable sites. Instead of showing you all 35 sites, I'm just showing the ones where we saw any variation. And we see two patterns. First of all, we see that, we see three patterns. The interesting thing is here in Photinus, there are no substitutions. So if it is resistant to cardenolides, it didn't do it by modifying this gene. The second pattern is that Photurus has gone and duplicated sodium potassium ATPase, and it's done that early on so that multiple species have this duplication. And then it's gone and made substitutions to one copy, but not the other. And we can show by ligand docking simulation, what we're doing is we're doing ligand docking simulations to the estimated native structures of the protein, binding the ligand to the pig copy, Photurus copy 1 in red, Photurus copy 2 in blue, and you see that resistant copy is displacing cardenolides from the binding pocket. And then finally, my original PhD training is in Drosophila, so I have kind of a special love for the organism. How do I work Drosophila into this story? Well, the thing is that all these insect species, even the vertebrates, they're going to be very difficult to study in any meaningful way in terms of assigning function to individual amino acid substitutions. But that's where Drosophila melanogaster comes in. Using state-of-the-art genome engineering techniques, we can actually make very slight alterations to the genome, we can go to the native sodium potassium ATPase locus here and just engineer any substitution that we observe in an insect of interest. And so I guess my eventual vision for this project would be something like this, where I could make a library of melanogaster strains that represent the type of genetic variation we see in these populations, and this might be a good framework for trying to understand the assign function to individual substitutions, and try to understand, for example, how groups of substitutions might be different than individual substitutions, this type of thing. So these are the people who did this work. Ying Zhen is a postdoc, was a postdoc in my lab. She just left. She spearheaded all of the RNA-seq and did the initial de novo assemblies. 2 graduate students in my lab. Matthew Artema did all the collections and phylogenetic analysis, and got in there and did the firefly RNA-seq. Molly Schumer did the really nice gene expression analysis just as she arrived as a graduate student. And then there's Edgar Medina, who's a student I met at Uni de Los Andes, but is now a graduate student at Duke University, and Dario Gersi, who was a postdoc with Mona Singh at Princeton, and they did the ligand docking simulations. And so that's it.
Well, I have a, well, go ahead, I have
Sure, thanks. So in a previous paper you showed that, I think it was in a coleopteran, that there were 2 consecutive substitutions, one and then the other, like, and it was repeated, I think, sort of deterministic. Is it still holding true, and what does it mean?
Yeah, so that's actually what I alluded to, I mean, cryptically here, is that I think we could understand interactions between substitutions better if we could control them in this way, in a controlled genetic background. But we have done this phylogenetic analysis where we see correlated evolution between substitutions. And in fact, the functional data says that if we move back to these frogs, for example, the combination of this substitution with this substitution is 100-fold more resistant to the toxin than individual substitutions. So there's very clearly some sort of synergistic interaction between those substitutions, and so we think that this is occurring. We saw it twice. Once in Lepidoptera and then once in Coleoptera.
And the first mutation is in the binding pocket or somewhere else?
No, they're both in the binding pocket, and they're somehow interacting, but we don't really know how.
There's similar stuff going on in hemoglobins, I think.
Yes, no, that's right. But I wouldn't doubt, once we can do these detailed functional studies in Drosophila, I wouldn't doubt that there's quite a bit more epistasis there that just has gone undetected, because I think the methods that have been used so far have been pretty crude. I think we can improve on that.
Is there also some interesting parallelism going on in the production of the toxins, or are the precursors just too distantly related to understand, to have that be any help in understanding constraints on adaptation?
Yeah, it's interesting because when you look at this compound, you think looks like a natural thing for an animal to make. So in retrospect, I think it's really surprising that the plants make it. How do they make this compound that looks kind of like a cholesterol? They don't naturally have it. But apart from that, I mean, I think nothing is known about the actual pathway for making these compounds. I think it might be pretty hard to get at. But this is not the only situation where you see convergent evolution for the evolution of this in plants and animals. This is one of a number of examples. So yeah, I think that would be interesting to look at eventually.
So Peter, there's a lot of power in the system because you have a specific target, and so you can look at this gene in lots of different things. but it's also kind of a candidate gene approach, right? You started with this gene, and so what can you say about the possibility that there might be other genes involved in dealing with the toxin? So you had one example where there were no mutations, and so I mean, do you worry that you're missing part of the story about the constraints that might exist, or other solutions that might exist?
Yeah, I mean, what we can say at the moment is when were substitutions at sodium potassium ATPase part of the solution, and when were they not part of the solution?
Yeah.
In fact, but actually, we're still missing potentially the regulatory variants. Like one simple way to achieve resistance might be to just upregulate the protein with no amino acid substitutions, soaking up the toxin with more protein. If you did that in a tissue-specific manner, maybe you'd be okay. And so we're kind of onto this, especially now that we found differential expression of copies. We're now looking at differential expression. But we made RNA-seq libraries basically to sequence one gene. But the data is still there. We're looking for correlated genomic changes. We can look for, and we have candidates. For example, anything that moves sodium or potassium around in the cell, even calcium or things that are linked to this. We have those phosphorylated glycoproteins and the generic ABC transporters. What are those doing in the transcriptome in apocynases specialists versus non-specialists? And that's another avenue. We're approaching this in a more holistic genomic way. But this is satisfying because we can really get at, this substitution does this. Whereas when we find something from these genomic studies, it's going to open up a lot of things to do.
Yeah. So, but related to that, I mean, specializing on something like milkweed probably involves a lot more than just detoxifying or dealing with the toxin that exists.
Absolutely, yeah.
And so, are you thinking about all the other convergent adaptations that might exist, like both attraction to chemicals or being able to digest whatever is in specializations for feeding on milkweed, that sort of thing?
No, I mean, I just think that is where single organism, single adaptation stories might give us at least a clue to how, just tackling that in over 300 million years of evolution is probably not a good way to go about answering that question. So.
Is there any evidence for any kind of co-evolutionary arms race between plants and insects in this system?
Oh yeah, no, absolutely, yeah. So on the plant side of things, which I don't study, but I collaborate with Anurag Agarwal, who's mainly in on the plant angle here. Cardenolides, it's not just one compound the plants are producing, they produce quite a library of cardenolides, and they diversify these things. When the plants are injured, they actually up the amount of cardenolide they add, this type of thing. Even these guys that are sequestering cardenolides, when they absorb them from the plants, they'll make additions and subtractions from the functional groups. And so this implies that this is in flux. And the plants also have a number of other adaptations against herbivory that are coupled into this as well. Like the cardenolides, milkweeds have a milky sap, and that's actually deterrent number one. As soon as a small insect takes a bite out of a plant, it just gets a mouthful of this latex, which has a good chance of killing it. So cardenolides are just part of the story.
So another one. So you probably know this paper that does genome-wide sequencing or genome-wide comparisons on the echolocating mammals, right? The bats and the dolphins, and if I remember well, they look at molecular signatures in parallel, and then they have this flurry of candidate genes involved in echolocation. Sounds mind-blowing. I don't know. What do you think? Do you think it could apply? Is it valid, or it seems like it?
Yeah, that paper kind of surprised me a little bit, because I feel like, first of all, that paper looks at a very specific form of parallel evolution. It's not going to have power to detect parallel recruitment, for example. It's looking for the same exactly the same substitution. So that study is going to miss a lot of parallel evolution, and yet they're saying something like some insane fraction of substitutions are parallel substitutions. If you add to that the number that you think you're missing, I think it would be more than, it'll be like 100% of substitutions or something like that. So I feel like something's wrong with that paper. But, that's the personal feeling. But there's not really they can't really relate it too much to phenotype. It's just by association.
But it might be able to harvest some low-hanging fruit in other genes, or I don't know. Yeah.
Oh yeah. No, I see what you mean. Yeah. We might be able to pinpoint it. We are looking for signatures like that. A certain PGP protein that always appears and is expressed in the right tissue at the right time. So there are more axes than just protein evolution.
Good. I just want to remind the graduate students that there will be pizza lunch with Peter in